learning to fold membrane proteins. So first, I will briefly introduce some background, like the contact prediction and the contact assistant protein folding. Then I will talk about our deep transfer learning method for membrane protein contact prediction, and then describe how to do the contact-based membrane protein folding. And finally, I will describe our results. So in the last talk, we know the membrane proteins are very important for drug discovery, but unfortunately, it's very hard to experimentally solve structures for membrane proteins. There are about 3,000 solved membrane protein structures in the PDB, but if we do clustering based on sequence, there are about only 500 non-redundant membrane protein structures. And so we also did some study on human genomes, and we found that there are about between 30 to 40% of human protein coding genes. They are predicted to be membrane proteins. Since in the PDB we don't have many structures, membrane protein structures, so homology modeling has very low coverage for membrane protein structure prediction. And the popular fragment assembly method doesn't work very well, especially for large proteins. And recently, the emerging method, the contact-based folding method, now is making very good progress. And so now we can use a contact-based method to do visual folding for very large proteins. But the key is to predict the contacts very accurately. So co-evolution method is the popular method for contact prediction. The basic idea is if two residues in contact, then it's likely they like to co-evolve. And so on the other hand, if we find two residues that are co-evolved, then it's more likely these two residues they are in contact. So this is the intuition for co-evolution-based method for contact prediction. Unfortunately, the co-evolution method works only on some proteins with a very large number of sequence homologs. If you don't have many sequence homologs, then usually the co-evolution method usually fail. And many membrane proteins, they don't have sufficient number of sequence homologs for very accurate co-evolution analysis. So we need to introduce other information to improve the contact prediction for membrane proteins. The co-evolution method only look at the pairwise relationship between residues. But actually, if we look at the contact map, we can easily see there are some patterns in the contact map. So look at this picture here, this example, we can see, so the contact map, the contacts in the contact map, they are not randomly distributed into the contact map. So there are some patterns, like for the coil region and helix region, so we can see some contact patterns. So we wanted to make use of this kind of contact patterns to improve contact prediction. So this is the intuition behind our algorithm. So in the last year, we developed a deep learning method for soluble protein contact prediction. So our deep learning method can exploit very high order residue correlation, which is also the contact occurrence patterns. And then our deep learning method can learn from thousands of protein family instead of a single family. So co-evolution method only use information in a single family when do the contact prediction. But our deep learning can learn from simultaneously learn from thousands of protein families. So can learn this kind of contact occurrence pattern to significantly improve contact prediction. So we formulated the contact prediction problem similarly as the pixel level imaging classification problem, which means we predict all the contacts of a single protein simultaneously. So this is very different from existing supervised learning method. So existing method mostly predict individual contacts one by one. So by simultaneous prediction of the whole contact map, then we can easily make use of the contact occurrence pattern. So we tested our algorithm in CASP12, and when we participating in CASP12, we haven't fully implemented our algorithm, but even so, in terms of the F1 score, our deep learning method still is the best. So on the 38 free modeling targets in CASP12. So this is the test result on cusp targets, but we know most cusp targets, they are not membrane proteins. So we wanted to study how well our deep learning method will work on the membrane proteins. The challenge for deep learning method on membrane proteins is usually we know deep learning method needs a large number of training data. So if you don't have a large number of training data, then deep learning you couldn't train deep learning model very well. And unfortunately, as I said before, in PDB we only have about 500 non-redundant membrane proteins. So we don't have many solved membrane protein structures to train our deep learning model. Then how to solve this problem? So we come up with a solution. So we use those non-membrane proteins. We know in the PDB we have about now 8,000 maybe or 9,000 non-redundant non-membrane protein structures. So we can make useful information in non-membrane proteins. So we use non-membrane proteins to train our deep learning model, and then apply deep learning model trained from non-membrane proteins to do the contact prediction for the membrane proteins. So this is why we call our method called deep transfer learning. So we transfer knowledge from non-membrane proteins to membrane proteins. Then why the knowledge learned from non-membrane proteins will work for membrane proteins? So we did some statistics. So this picture shows you the contact patterns, the similarity of contact patterns between non-membrane proteins and the membrane proteins. So look at here. So this shows you the, so we look at 3 times 3 small matrix, submatrix in contact map. Then we just do a statistics, and we pick up those highly frequently occurring submatrix. And so these top 20 patterns are from the non-membrane proteins. And these 20 patterns, they are from membrane proteins. So you can see they are quite similar. So they're only different in the ranking orders. But if you look at the top 20 patterns, they have a big overlap, which means the non-membrane proteins and the membrane proteins, they share some similarity in terms of the contact occurrence patterns, although the sequence may be very different. But their contact patterns, they are very similar. And this, in the bottom, we look at the 4 times 4 matrix, and we obtained the similar results. So this is why, this is the intuition why deep learning, when trained on non-membrane proteins, they will also work on the membrane proteins. Now I'm going to introduce some details about our deep learning algorithm. So our deep learning algorithm will start from the multiple sequence alignment. So from the multiple sequence alignment, we derive 2 types of features. One is called sequential features. So sequential features just like the sequence profile and the predicted local structure. And then we also have pairwise features. The pairwise feature including the mutual information between 2 columns in the multiple sequence alignment, and also the direct convolution information between the 2 columns in the multiple sequence alignment. So we fit these 2 types of features into our deep learning model, then we will predict one contact map like this. So this is the input and the output of our deep learning model. And here is the architecture of our deep learning model. So here, starting from the sequential feature, we do the convolution transformation. We do the convolution transformation about 10 times. Then we convert the sequential feature into a pairwise feature, and combine the original pairwise feature with this converted pairwise feature together to form a new pairwise feature. Then we feed this new pairwise feature into the 2D convolution transformation. And then finally, we can predict the contact map like here. So the 1D convolution transformation is used to capture the sequential information, capture the sequential context of one residue. And the 2D convolution model is used to, the 2D convolution network is used to capture the 2-dimensional context of a residue pair. So this is our model, and for details, you can look at our paper. And here there are also some details about the networks. So here, so each block consists of some layers. So like the activation layer and the convolution layer. So convolution layer do linear transformation, and activation layer is used to do nonlinear transformation. So by combining them together, then we can model the nonlinear relationship between the input and the output. So this is the detail of our deep learning network architecture. And then we also experimented 3 different strategies to train our deep learning model. The first strategy is we just use the 500 memory proteins to train our deep learning model. So we do 5-fold cross-validation. The second strategy is we only use the non-memory proteins to train our model. So we remove redundancy between all the sequence similarity between our non-memory protein and the memory proteins depend by the sequence identity and also by plus E value. The third strategy is we mix non-memory proteins and memory proteins together to train our model. And in the third strategy, we also do the 5-fold cross-validation on the memory proteins. And we combine both the memory protein specific feature and also memory protein independent feature. So this is our training strategy. And then we evaluate the contact accuracy. So we use a very simple method. We just look at the top LK predict contacts to see how many of them are correct. So this is our accuracy measure. So this table shows you the accuracy. And we also compared our method with other methods. Like the top 3 methods, the first 3 methods, they are pure correlation method. And the metacycle with the supervised learning method. So metacycle takes a cycle and other pure correlation method as input, and then also using the sequence profile information and other features to do the prediction. And the MP is our deep learning method trained by only memory proteins. And the non-MP is our deep learning model trained by only non-memory proteins. And this is our deep learning model trained by both non-memory proteins and the memory proteins. So from here you can see, when we look at the medium range or long range accuracy, so our deep learning model trained by only memory proteins, they outperform these first 4 methods, outperform metacycle and outperform the pure correlation method by a very large margin. So we just trained by memory proteins. But if we look at this, our method trained by non-memory proteins, and it's even better. So although we don't use any memory proteins to train our model, but the performance accuracy is better than the model trained by memory proteins. So which means the non-memory proteins contains important information for memory protein contact prediction. And then if we mix them together, then we can even improve the accuracy of the contact prediction. So the best way is to mix the memory proteins and the non-memory proteins together to train our model. So this is the contact prediction accuracy, and then we feed the predicted contacts into CNS as software package for the NMR's data analysis. So we feed our predicted contacts to a CNS to build the 3D model. Then we can evaluate the quality of the 3D model by TM score and to see how well we can do the 3D modeling by using the predicted contacts. Again, so here we compare. So these left 2 figures compare our method with the CCM plate, so which is one of the best pure correlation method. So this picture shows you the comparison between the mixed strategy with the CCM plate, and this shows you the comparison between the model trained by only non-memory proteins with the CCM plate. And this picture shows you the comparison between our strategy and the metacycle. So each point in the figure represents 1 protein. When pointed lies in this region, which means our method is better than the other method. So you can see tested on the 500 memory proteins. So on most proteins, our method can generate significantly better 3D models. So this is the comparison with the CCM plate and the metacycle. And here I also have a table to summarize the 3D modeling accuracy on these 500 memory proteins. So here again, I compare with, I also listed the performance of CCM plate and the meta curve. And here I can also compare with the homology modeling. So like here, when I use the non-membrane proteins as the template database to do homology modeling, and here it means I only use the membrane proteins as a template database to build the homology modeling. And these three are our contact-based modeling method. So look at the average TM score. So you can see here. So when you use only non-membrane proteins in the template database, then the result is very bad. So the average TM score is only 0.17, which is the expected score for any two unrelated proteins. So which means that the result is just random results, which also means there's no redundancy between our non-membrane proteins database with the membrane protein database. And then when we use the membrane proteins as the template database, we can build some good models, although the average TM score is still low. And when we use the membrane proteins to train our deep learning model, then we can get a better TM score than using the membrane proteins as the templates to build the template-based modeling. Then if we only use the non-membrane proteins to train our model, then we can get a significantly better TM score. So the TM score is even better than when the model is trained by membrane proteins. So this means, although the non-membrane proteins are not very useful for template-based modeling, but the non-membrane proteins are very useful for contact-based application folding of the membrane proteins. And if we mix them together, then we can improve the TM score. So this is the average TM score. And then we can also look at the RMSD, the average RMSD. So you can see when we use only non-membrane proteins to train our model, we can get the RMSD is pretty close to the RMSD of the model trained by both non-membrane proteins and the membrane proteins. And then we also did some statistics, like we count the number of models with TM score with RMSD less than 3 angstrom, and with RMSD less than 4 angstrom, and with RMSD less than 6.5 angstrom. You can see when we use this criteria, then we can get more than 30% of proteins can be folded with RMSD less than 6.5 angstrom. And if we use TM score as a criteria, like if we use TM score 0.5 as a cutoff to determine if a fold is correct or not, then we can see more than half of them has the correct fold. So we can predict correct fold for more than half of the membrane proteins by using the deep learning model, which is much better than the template-based method. So this is our self-test on the 500 membrane proteins. And we also test in the Camel, do the blind test. So Camel can be interpreted as a fully automated cusp. So it's a null benchmark. And when we do the test, no one has the ground truth. So each weekend, Camel will send us some sequences and we do prediction. And here I show you some examples, like this for this example. And so this protein has 210 amino acids, and it has more than 1,000 similar sequence homologs. But even for protein with so many sequence homologs, when you look at the contact prediction accuracy, you can see our method performs much better than the other methods, especially much better than the pure correlation method. So even for a protein with 1,000 sequence homologs. And then this is the 3D modeling accuracy. So look at the TM score here. So we can get TM score close to 0.9, and the RMSD is close to 2 angstrom for this protein. And then look at this is the low zeta server. So the RMSD is 5 angstrom, and the TM score is only about 0.7. So we generate a much better model than the low zeta. Low zeta uses a pure correlation method to do the contact prediction and then do the folding simulation. But we don't do any folding simulation. We just feed our predict contacts into CNS. So this is one example. And here is another example. So this is the example. It even has many more sequence homologs. It has 7,000 sequence homologs. And from here you can see, in terms of contact prediction accuracy, we still do better than the other method. And in terms of 3D model accuracy, we are better. We are slightly better than low zeta, although not much better. So this is in terms of the 3D model accuracy. So for protein with so many sequence homologs, we can still do better. And this protein now has only about 100 sequence homologs, so it's very hard for pure correlation method to do the contact prediction for this kind of protein. So you can see the accuracy here. So the accuracy for the pure correlation method is very low, but we can do much better by using the deep learning method. And this is 3D modeling accuracy. Now you can see with the best model we got has a TM score 0.6, but the Zeta has a TM score only about 0.4. So we can get the correct fold, but the Zeta failed to produce a correct fold for this protein. So this is the Kamiya test targets. Okay, finally, I will just summarize my talk. So this is the take-home message. So deep learning can significantly improve the contact prediction for both soluble and membrane proteins. And the deep learning model trained from non-membrane proteins can improve membrane protein contact prediction. So non-membrane proteins are not very useful for homology modeling, but very useful for the contact-based application folding for membrane proteins. This is because the homology modeling most depends on the sequence similarity, but our deep learning model depends on the make use of the contact occurrence pattern instead of the sequence similarity. So this is why the non-membrane proteins are useful for our method. And finally, our folding algorithm runs is very efficient. So running on a single Linux workstation, it takes about 30 minutes to a few hours to fold one protein. Okay, yes, thank you so much.
So I'm actually going to propose that we have no questions now, because we are significantly behind time, and I suspect everyone would like to get to the posters and see everything else. So please make sure you ask Jinbo questions after his talk. I'm sure he'll be around for everybody. Thank everyone for being here, and can we again thank all the speakers who've spoken in this session and